Hi, Dr. Atchison here, and this is our fourth video in Chapter 2, and now we're going to be talking about retinal ganglion cells specifically. So this is a different image of the pathways that I showed you earlier. This is an image from your book. It's figure 216. And again, it's showing you the same thing that the other one, the RH bag showed you, so that we go receptors, which are the cones, H is horizontal cells, B is bipolar cells, A is amacrine cells, and R is retinal ganglion cells. So the RH bag, this is the same thing that we talked about in the photoreceptor video. And remember that the way that the information goes in is the light comes in this way, and the cones get that information, okay? So the cones are, in this picture, the rods and the cones, but here we only have cones, are our photoreceptors. That's our sensory receptor. That's where the information is going to. So the light comes in, passes all those cells, the photoreceptors capture that. The photoreceptors then take that information and send it back this direction, going through, again, the RH bag order. So receptors, horizontal cells, bipolar cells, amacrine cells, and then retinal ganglion cells. So again, we go through that order. And it's also important to remember that a process here is that many to one, okay? So we have 4 cones here, and we only have 2 horizontal cells and 2 amacrine cells. So we're going from many cells to 1, and 2 of the information's going in. And so we've got this kind of funnel system, okay, where we're getting many things and kind of going through one output. That idea of that funnel system is what's called convergence. Convergence is that many-to-one process. Because there's so many rods, they have to converge or share ganglion cells. So remember, our rods are primarily in our periphery, and so they're not on our fovea. They're out other places on the retina outside of the fovea. Because there's so many of them, remember there was 20 times as many of them as there were the cones, they're going to have to share those RGCs, okay? More so than the cones do. And this is, again, called convergence. This is where those retinal circles, multiple photoreceptors, in this case, mostly it's rods, mostly we have rods to have the most convergence, send signals to just one retinal ganglion cell, okay? And remember, RGC stands for the retinal ganglion cells. Typically, rods are referred to this many-to-one. Typically, they are the ones that have the most convergence, okay? Where cones have few-to-one or one-to-one convergence. So they may just be a couple of cones, share a retinal ganglion cell, or sometimes it'll be one cone to one retinal ganglion cell, okay? And again, this explains why those rods are so much more sensitive to low levels of light, why our cones are really about acuity, okay? And we'll talk about this some more on one of the next slides. But remember, if our cones are getting, and remember, our retinal ganglion cells is kind of that pathway to the brain, right? That's our direct, their axons go straight to our visual cortex. And so, through that optic nerve. And so if we have one cone getting information, giving information to one ganglion cell, that's really allowing this level of acuity that we get with the cones that we don't have with the rods. And again, this explains why rods are more sensitive to low levels of light, because there's so many of them collecting this information that they can detect that level of light, that dim light, and that work on the periphery so much better than the cones can. Circuits that have this convergence also have a property that's called spatial summation. And this is that the firing rate of that retinal ganglion cell increases, so it goes up, as the number of photoreceptors that are activated by light increases, okay? So the more, if we have this many-to-one process, okay, so say we have 4 rods going into this one RGC, the more of those rods that are receiving light, so if we have 4 of those rods receiving light, that's going to fire more. On that RGC is going to fire more than if only one of those rods was being stimulated. So again, this is kind of about the input in that that funnel. So if you just put a little bit into that funnel, you're just going to get a little bit out of that funnel. If you put a lot into that top of that funnel, you're going to get a lot more out of the bottom. So think about pouring liquid down it. If you're pouring just a little bit of liquid down a funnel, it's going to be more of a slow trickle, where if there's a whole bunch of liquid in that funnel, that pressure is really going to increase the rate in which that liquid is going through the funnel. It's the same thing. The firing rate of that RGC, that bottom of that funnel, goes up with the more light is being activated on those other cells. So again, this spatial summation, the definition from your book, is the property of the retinal circles with convergence in which signals from photoreceptors in some small space on the retina add up to the effect of this and the RGC. Okay, so again, the more pressure we have, the more light we have being received by more of these rods, the higher the fire rate of the RGC. And the degree of this convergence is affected by two different characteristics. Okay, so the higher degree of convergence, that allows for more dim light. So remember, if there's not much light coming in, if that light is hitting 5 photoreceptors and they're all going to 1 RGC, we're getting an increased firing rate, okay? Whereas if it's just going to 1 photoreceptor, we're not going to get that, okay? So again, it allows that higher degree of sensitivity to this dim light because there are so many of these cells to get them. This is one of the reasons that rods are so much better in dim light, is because there's so many of them. And this process of funneling them into fewer RGCs really increases the firing rate, okay? So this funnel process is what makes these rods more simple, better at detecting this dim light. The lower degree of the convergence, so if we have a few to 1 or a 1-to-1 convergence, this is about acuity, okay? This can give us more information about the details, okay? So if you have 1 cone getting information and it's sending it to 1 RGC, we don't have any convergence there. We have no convergence. But then we can get information about acuity. We can see really well. We can look at things very particularly. So here's a graph from your book. This is figure 224 on page 68. And if you look at the top left where it says A, that's the degree of convergence. So the high convergence, the one on the left that has the 4 cones going to 1 RGC, that's going to be a bigger funnel, right? It's going to have more cones in this case going to that 1 RGC. Where the 2 cells going to the 1 RGC, that's moderate convergence. And then when we have that 1-to-1, we have no convergence at all, okay? So when I was talking about that convergence, here's the difference. The more cells, the more photoreceptors you have going into that single RGC, the higher the degree of convergence, the bigger your funnel is, okay? Whereas the fewer, the smaller it is. We have, or in some cases, no convergence. And the way that spatial summation works is the top right, where it's talking about firing rate, okay? So on the left where it says low firing rate, you can see coming out of that RGC, we have less ticks than we do on some of the other cells. And that's because we're just putting a little bit into that funnel. Only one of those photoreceptors is being activated, okay? In the second picture, in the moderate firing rate, 2 of the 4 are being activated. And in the high firing rate, all 4 of them are being activated. So again, think about what's going into that funnel, okay? Either the funnel's the same size on all 3 of these pictures of B. What's different is how much is going into the funnel, okay? And so we have a little bit going into the funnel, so we have a low firing rate. We have a whole bunch going into the funnel, we have a high firing rate. So those are really the big things that I want to point out. Again, you can look at the acuity on C2, where we have that 1-to-1, we're going to get more output. Where we have no convergence, we'll have higher acuity than when we have high convergence, we'll have lower acuity, okay? Another thing I want to point out about these pictures, especially if you're thinking about looking at A, those highlighted areas around those cells, let's refer to those as receptive fields, okay? So that's how much receptive field is going into something, okay? And we'll move on and talk about those some more. So receptive field is the area that when stimulated causes a change in the firing rate of a single cell. Okay, so again, and that's going to be that RGC. We're going to have a change in the firing rate of that. And it doesn't say an increase or a decrease. It says a change. So either an increase or a decrease can work. So this will work for excitatory messages, and this will also work for inhibitory messages, okay? So it's just a change. Receptive fields really vary in size. Areas in the fovea have much smaller receptive fields than areas in the periphery. That's why you have so much better acuity in your fovea, is because, again, that is where you have smaller receptive fields. You have more cells firing than you do in the periphery, okay? We don't have as much convergence in the fovea as we do in the periphery. And different types of RGCs have different size receptive fields. And there's actually more than 20 different kinds of RGCs. We are not going into that level of detail. We'll talk about two here. And that was mostly for illustrative purposes, to talk about these differences of receptive fields, okay? So again, the cell's receptive field is this little piece of this whole greater picture that the eye is seeing. So the number of photoreceptors in a retinal circle, so kind of how big your funnel is, and the size of the receptive fields vary greatly within the position of the retina, okay? So remember, if you're on the fovea, you're going to have much smaller receptive fields than if you're further out, okay? If you're further towards the periphery, those are going to be much larger receptive fields. And the size of the receptive field really depends on the size of the RGC's dendritic tree, okay? So that's just how many dendrites does that cell have, okay? So that RGC, how many dendrites does it have, okay? And so here, all these different colors on this picture, this is a micrograph, and it shows the dendritic trees of 7 RGCs in a rabbit's retina, okay? So these are all different RGCs, and these are all their different dendritic trees, okay? The more dendrites it has, that means the more connections it has to make. The bigger the funnel, the bigger the receptive field. Which, remember, a bigger receptive field is going to be not good for acuity. We want a small receptive field for acuity. So here we have the more dendrites something has, the less acuity it has. But the better it's going to be at light and that kind of periphery information. So note here that the trees, and then therefore the receptive fields of the RGCs, tile this part of the retina. There's not many gaps between them. There's a lot of overlap between them with very few gaps. And this is good for vision. You don't want gaps in your vision. You don't want gaps in your receptive field. So notice here that there is a lot of overlap. So the two kinds of RGCs we're going to talk about are midget RGCs. And here in this image, those are the purple ones. And parasol, which are the larger ones, and those are yellow. And they both have different receptive fields. And the easy way to think about this is midget, smaller. Parasol, if you open up a parasol or an umbrella, that's going to be larger, right? So that's the way to remember which one's smaller and which one's bigger, is by their name. So on the left, we have the receptive fields of the parasols. Now, if we're looking at this picture, I want you to focus or foveate on the sphinx's face, okay? Let's put that on your fovea. That's what this is simulating, okay? Where those receptive fields are smaller, closer to the fovea, you see that. In both the parasol and the midget, we have smaller circles. Those circles are representative of the receptive fields. They're smaller towards the retina because that's where we've got more acuity. And they're larger as we go towards the periphery. So we can have both midget and parasol RGCs in the fovea and in the periphery. But again, their size will depend on where they are. In general, though, parasol have larger receptive fields and midgets have smaller receptive fields. And again, you have overlap between these. So the picture that's on the far right, we have the overlap between the purple midget and the yellow parasol, and that accounts for a lot more acuity, okay? Because we have all this overlap and getting simultaneous information from different receptors. Okay, so to continue on this receptive field, they found out that an American physiologist named Kuffler figured out that we have center-round receptive fields. And what this means is that the center of your receptive field responds differently than the outside of it. Okay, so I call this slide olives in your eyes, because to me that looks like an olive. Like if you're looking at an olive head on, it's a green olive with a pimento stuffed in. The pimento would be the center of your receptive field, and the olive itself is the surround. And what happens is when the pimento, or the center of your receptive field, is activated, it responds differently than the olive does, because they're different things. Okay, you can kind of think of it that way. In terms of your receptive field, it's all one, but those things respond differently. Okay, so again, the center responds differently to stimulation than the surrounding portion. And how it just responds depends on what kind of receptive field there is. Okay, there's two different ones. On center and off center. In this graph here, we have the on center is on the left and the off center is on the right. And these are the firing rates of different stimuli. One of your assignments for this week is a Launchpad assignment going through this, and it allows you to play with bright light, dim light, and background on both on-center and off-center receptive fields, and helps you figure out the differences in firing rates, how these receptive fields work, and how these olive-shaped receptive fields work out. So do that Launchpad assignment. That will really help you with this understanding. I'm not going to lecture on it because of the assignment. Does such a good job. Okay. I do recommend at this point pausing the video and coming back because it really will help you understand the rest of the lecture if you've done this receptive field video activity prior to moving on. Pause, come back. Okay, let's assume that you've all done that. Edge enhancement. Okay, so what happens in this is a little bit of a perceptual, just lost my word. A perceptual illusion here. These are called Mach bands. And when you look at it, it looks like each color, each gradient, like the dark versus the light, is kind of a little bit of a gradient in and of itself, where it seems darker on the left and lighter on the right. The reason of that, the reason of that is that perceived intensity. So if you look at that perceived intensity underneath it, that's what it looks like, where it's a little bit more intense on the left of that color field than it is on the right. In actuality, each of those colors, if you look at the physical intensity, is the same level of intensity throughout. The reason that it seems that this is almost a series of gradients is because of edge enhancement. Edge enhancement is the process in which the visual system makes edges as visible as possible. And it really facilitates our awareness, our perception of where one object and another object end. Okay? And this is really important. I mean, if you think about engaging in the world around you, knowing where the edges of things are is one of the most important jobs of our visual system. And so because it's so important, the visual system tries to give you a leg up and kind of enhances those. So when we looked at this image, those edges are enhanced. And that's what's creating this illusion of the difference in perceived intensity on the edges. It's because, again, our visual system is trying to say, no, look, there's an edge there, pay attention. And so it's highlighting it for us. It's enhancing it for us. And partially this is done through those center surround, our olives, receptive fields, okay? This example is figure 230 in your textbook, page 75. And these are all, in this example, are all on-center receptive fields. You could do it with off-center, but this one happens to be on-center. And so remember, the orange centers are responding differently than the green surrounds, okay? And so where we have these two receptive fields right on the edge, we're getting information from RF1 and receptive field 3, RF3, that are saying, hey, there's an edge here, and they're both telling you this. You can see one of which has a more decreased rate of firing, and the other one has a more increased rate. And then you look at RF2 and RF4, and they have a very similar rate of firing. So again, because those have a similar rate, they aren't enhanced. Whereas we look at this difference between RF1 and RF3, and it's really saying one is more inhibitory and one is more excitatory. And so it's saying, look, there's something going on here. There's an edge. You need to pay attention. So again, these receptive fields can really help our perception by heightening things that we really need to pay attention to. So that concludes our coverage of Chapter 2. I'm not going to go into the visual disorders that are covered in your textbook. If you're interested in it, by all means, read, but you will not be tested on the disorders of vision. Thanks, and I'll see you again.